Hi students, in this video I am going to explain you about the sexual reproduction of the flowering plants, and flower is representing the sexual reproductive organ and how it is formed, all the things I discussed in the previous video. Now in this sexual reproduction of the this one in the flower and flower is the unique characteristic of the angiosperms also. It is a sexual reproduction, it is doing. Sexual reproduction is a lengthy process which contains of so many stages. So this lengthy process is divided into two parts. One is the pre-fertilization structures and events. Means sexual reproduction means zygote formation. Male gamete and female gamete used to fuse and forms the zygote. Up to there, so many events are there. All those things will come under pre-fertilization structure and development. Next part is the zygote will develop as the embryo and forms the seed. That is post-fertilization changes. Now we are going to see pre-fertilization structures and events. So here the first flower has to be formed because both the male and female sex organs are present inside the flower. So here the where the flower used to present on the plant will be decided. The actual flower formation place on the plant is decided much before they appear. Means whatever you are seeing, we are seeing, we may think, but the thing is before, so many days before only it used to that it will, the formation place will be decided how any type of the organ in the organ formation in the plant will be done by the meristematic tissue. Meristematic means they continuously dividing tissue. So here the meristematic tissue, apical meristem. Apical meristem means not shoot, apex, root, or the, it will be the whatever the axillary bud apex, apical meristem, or shoot apical meristem. What are the apical meristem will be take the decision where the flower has to be formed, and the apical meristem will be converted into floral primordium. Floral primordium is nothing. It is a meristematic tissue which is converted from the apical meristem. The apical meristem convert into floral primordium. Floral primordium is going to produce the flower or inflorescence. That is the important point. And all these things will be done by the because of several hormonal and structural changes. Floral primordium produces flower or inflorescence. This is most important thing. This floral primordium used to produce either flower or inflorescence. Both will be produced by the floral meristem. The need, it is important. And the flower used to contains of male and female sex organs. Then the male sex organ in this, that is the andricium, consists of stamens, consists of contents of stamens, and the female sex organ, that is the carpel or pistil we used to call. And how they will develop from the floral primordium? Floral primordium is a meristematic tissue that will produce the permanent tissue. The permanent tissue will be differentiated as the sepals, petals, and stamens and carpel. So the flower male reproductive structure andricium and female reproductive structure develops from the differentiation of the floral primordium. Here, whenever we are seeing here the floral, the main thing what you should remember here is that so hormonal, and because of the hormonal and structural changes, it produces the flower or influence. Which one? That is the floral primordium. The flower male reproductive structure andrecium and female reproductive structure gynoecium develops from the differentiation of floral primordium. Next, andrecium consists of the stamens. Already I told that the stamens and the gynoecium consists of the pistil or the carpels. These are the how the male and female sex organs are forming and the flower is developing from the meristematic tissue he given in one paragraph of your NCERT book. Now here we'll see next thing, that is the mandricium is represented as the stamen, and stamen consists of the microsporangium. Microsporangium used to produce microspores are nothing, they are the pollen grains. These things we are going to discuss in this video. So here the thing is, stamen consists of stalk or filament, and terminally it is a bilobe anther. It is to have the one filament, and it is to have the one stalk, that is the filament, and terminally it is to have the anther. The number and length of the filament variable in the different species. The stamen number or the length of the whatever the filament is to vary in the species to species. And here you should remember one thing. The thing is filament length. Whatever the lengths are, different types of the filament lengths are present in the same flower. What is the example? That is in your book, go and see. That is called as tetradynamous condition. In the cruciferous family, recently he added tetradynamous condition, different lengths. He may connect this and that one for the need, remember it. And this and the variable different species. Each and next, whenever we are seeing the terminally, it is having the bilobed anther. So bilobed anther, each and lobe used to contains having two theca, which contains two microsporangium. And now we'll see the diagram. This is the stamen. It is having the filament and anther. And the anther generally used to contains of the two lobes, and each lobe used to have the two theca. These are the theca. And in between the two theca, there is a longitudinally a groove used to present. And that groove, which is to separate the theca, longitudinal groove. And how many grooves used to present in a stamen? Two. Because in this lobe one, that lobe was a one. And next, whenever we are seeing, the anther is mainly it is mainly it is producing the pollen grain. So whenever we are seeing the anther transfer section, it used to have, this is the one lobe and this is the second lobe, and the each lobe consists of the two theca, and longitudinal groove will be used for the dispersal of the pollen grain. It used to break and send out the pollen grain, that he has shown in this one. Now whenever we are seeing here, each lobe of anther having two theca, which contains two microsporangia, that already shown. A longitudinal growth separates both the theca, and Here the anther is tetragonal because it is the, you see here, tetragonal, 1 2 3 4 4 4 whatever the we are seeing that it is having the four sacs at the four corners, that is theka, four theka. That's so it is called as a tetragonal, 1 2 3 4, and each is present in the corners. Different way. Tetragonal structure consists of the four microsporangia. This is the microsporangia. So one globe consists of the two microsporangia. The first it is called as a theca. Theca is a part of the lobe. Each lobe consists of the two chambers called as a theca, and the theca consists of one microsporangium. Consists of the microsporangia in the corners you see there. And further development of the microsporangia becomes a pollen sac. Now the whatever the what here we are seeing microsporangia, whatever the anther, tetragonal and microsporangia. Microsporangia will be converted as pollen sac when it is after maturation of the microsporangium, it produces the pollen grain. So entire that what a microsporangium, which is the longitudinally having the groove, and it is completely filled with pollen grains, which are in the maturity condition before dehiscency. Dehiscency means breaking of the. It used to break because all my pollen grain are present inside. They must come out by the dehiscency. So at that time we used to call, when mature pollen grains are formed, then it is called as pollen sac. Before that one we used to call it as microsporangium. Before that one we used to call it as a theca. Theca, theca will become microsporangium, microsporangium will become pollen sac, and the pollen grains are formed. This is the sequence because he is giving the sequences also. Now whenever we are seeing here, the further development of microsporangium becomes pollen sacs with pollen grain. This is given in the NCERT book also. Now we'll see the structure of microsporangium. So microsporangium is important for the producing of the pollen grains, and without pollen grains there is no question of sexual reproduction. So structure of the microsporangium is important. The microsporangium appears near circular, almost circular shape it is to present. Outline it is the circular. It is surrounded by the four walls, that is the epidermis, endothecium, and middle layers, and next is the tapetum. Microsporangia appears nearly circular. It is surrounded by the four walls, layers, wall layers, as epidermis, endothecium, middle layers, and tapetum. Here also some need bit is there. Here you see epidermis in the diagram clearly has shown, that is one layer only, one layer of epidermis, one layer of the endothecium, and middle layers more than one layer, and inside fourth layer wall layer is tapetum. And all the three layers, whatever are that is the epidermis, endothecium, and middle layers, they will be useful for the protection purpose of the sporangium. This is the sporangium. Here the sporangium will be protected by how many walls, he can ask. three wall layers. What are they? Epidermis, endodermis, and middle layers. Out of that one, which contains of with single layer of cells, and which contains of many layers of the cells. Many layers will be the middle layers. This is most important bit for the need. Remember it. And tapetum is mainly meant. It is the biggest cells which is to present around the central tissue. That central tissue, whatever is there called, we used to call it as sporogenous tissue. Means which is to produce the spores. That is called as sporogenous tissue. It used to present in the inside of the sporangium, microsporangium. And microsporangium is covered with four walls: epidermis, single wall; endodermis, single wall; middle layers, more than one layer it is to present; and tapetum. Out of the three, epidermis, endothecium, and middle layers will involve in the protection and dehiscence of the anther also, because here it is breaking and sending out the pollen grains through the by the dehiscency, means by breaking. That will be decided by which layers, means outermost three layers, that is the epidermis, endothecium, and next will be the middle layers. Now the thing is, the fourth layer from the is tapetum. Tapetum consists of the big cells and thick, very thick cytoplasm, and more than one nuclei is used to present in that. This is asked in the 2025. Tapetum cells contains of the tapetum cells is given, and it is to have more than one nuclei. That is correct assertion. Reason he has given, the reason now I will discuss, that is it is having the more than one nuclei. Means why the more than one nuclei is present, how it is formed, he has given in the textbook. But the thing is, because of the plasmogamy means only the two cells which to fuse together, that is plasmogamy, karyogamy cannot be taken. So two nuclei will be separated, but the two cells will become by the plasmogamy single cell. That what is the advantage? That I used to discuss from the last 10 years in my class, but it is not given in the textbook, but it came in the NEET as the reason. Efficiency of the tapetum will be increased by the more than one nucleus by having this one. Why? Because one nucleus is there, genes will work for the one nucleus, whatever they do, it will do. Two nucleus are there, double work it will do. Means why? Because the tapetum used to nurses the water sporogenous tissue. Means whatever the pollen grains will be developed from the sporogenous tissue, which are the center, and it will be nourished, means it will be feeded, means it will give the food material will be supplied from the tapetum. Tapetum is having the more than one nuclei to increase its efficiency. So assertion is correct, reason is correct, explanation is also correct. But majority people unable to understand the reason. One more example I am telling here, that is, all of you know, that is the whatever the endosperm, double fertilization, triple fusion, it used to have 3n number. That is also advantage, that I will discuss there. So here, but in the diagram he has shown one, wrongly he has shown that is one nucleus only, but it is having the two. More than one means two or three, whatever it may be. But diagram has shown one only. That we will see. And now it is, this is one lobe and this is the second lobe. Each lobe is having the two theca, dithecus, and each theca consists of one sporangium, and this sporangium is covered by the four layers. That is, the four wall layers are the epidermis, endothecium, middle layers, and tapetum. And next, whenever we are seeing outer three layers are useful for the protection purpose, and the tapetum used to present fourth layer and biggest cells. They used to have the thick cytoplasm, and they are having the more than one nuclei. This he has given in the 2025, means you can understand in which way he is asking the question, and its importance also he is asking. Now you see here, and next, whenever we are seeing the microsporangium appears near circular outline, it is surrounded by the four wall layers as epidermis, endothecium, middle layers, and tapetum. The outer three layers performs the function of protection and helps in the deficiency of anther. Means here, where the deficiency will be taken in the long groove, whatever is present in between the two sporangium, that is the tooth theca. The innermost wall layer, tapetum, contain dense cytoplasm with more than one nucleus. This he asked in the 2025. So, why it is also discussed. Now, it nurses what the tapetum nurses the developing pollen grains. Means for developing pollen grains required more food, because in short time it has to develop, so lot of food material is required for the development producing the pollen grain. So it will be done by the tapetum. It used to give and it used to absorb the food material from the plant, mother plant. So the nurses development pollen grains. What is the next is the center of the microsporangium that I already shown. That is the microsporangium occupied with sporogenous tissue used to present in the center. Now we'll see the microsporogenesis. How the microsporogenesis will be taken. Already that is the sporogenous tissue where it is present. Now you see once more. Here you see clearly sporogenous tissue is to present inside the tapetum. Remember it, you can ask, because all are the statements. For that only I am showing you clearly. 6 questions given last time, but no question. That generally most of the 3-4 questions are testing type. And one more thing I am telling you, the persons who are going for the 2027 CBSE exam, in that one also 16 one mark questions he is giving, and 3 marks questions, all other questions also testing type only, concept orientation they are asking. If you don't know properly the concept, you cannot get more marks. You will get the 60% like that, but if you want to score more than 90%, you have to follow all the things. The sporogenous tissue is surrounded by, he will give, that is the sporogenous tissue is covered by the tapetum, outer the tapetum, middle layers, outer the middle layers, it is having the endothecium, outer the endothecium, it is having the epidermis. And now what happens? Here the sporogenous tissue used to present inside the tapetum, and it is have already the plant angiosperms are the diploid, the 2n number. So it will take meiotic division, meiosis. Meiosis division means what? The Meiosis already you know that in that one, meiosis gives the 4 daughter cells, and each daughter cell will have the haploid condition. Haploid condition it will come. Why it is, how it is, all the we discussed already in the cell division. So meiosis will be taken place in which of the cells? In the microspore, that is the sporogenous tissue. The sporogenous tissue, each cell that is the will work as spore mother cell, microspore mother cell, means which is going to take the meiotic division, meiosis. So that entire whatever the cells are present in the sporogenous tissue will act as microspore mother cells. Each cell will take the meiotic division and forms 4 haploid spores. That is called as microspore tetrad. 4 spores are present, so it is called the tetrad. And the microspore tetrad means meiosis has taken meiosis 1 and meiosis 2, and 4 cells are formed, and there used to present cluster, means all 4 are not separated. But at the maturity of this anther, it will be dehydrated, means water will it lose. Then it will be separated, means the size of the spore also will be reduced and they will be separated. So then it will produce the 4 microspores, and microspore further develops as the pollen grain. Pollen grain development we will see in the next video. Now you see here, and after the pollen grains are formed, matured, it will be sent out through the decisions of the anther. Already we discussed. Now you see here again once more. It is surrounded by the 4 wall layers and epidermis, all these things. Out of 3, it is the protection, 3 outer 3 will give, and the inner most layer the tapetum, and it used to dense cytoplasm, and this is asked in the last year, the question. And nourishment, another thing, the center of microsporangium occupied with sporogenous tissue. Now we'll see the sporogenous tissue, how it will produce the microspores. And in this one already I discussed, that is the each cell of the sporogenous tissue undergo meiotic cell division and forms microspore tetrad. Each, now in the diagram is shown, that is the microspore mother cell. Each is microspore, that whatever the sporogenous tissue, whatever the tissue is in the tissue, whatever the cell is there, it will work as the spore mother cell, and it will take the pollen, meiosis, and forms the 4 microspores, microspore tetrad. When anther mature and dehydrate the tetrad, and the anther when it is matured, it will be dehydrated, it loses the water, and the tetrad separated and gives 4 microspores. So now here meiosis he told, and meiosis used to form 4 cells, all are the haploid cells. So all are the haploid microspores are formed, and the microspore develops as pollen grain by the time of the decisions of the anther, because the microspore is formed by the meiosis, by taking of the meiosis by the spore mother cell, which is present in the sporogenous tissue, and this the whatever it is the haploid cell which is formed by the meiosis of the microspore mother cell will develop, and further development it used to form 2 it is to take further divisions and forms so many cells, that it will come in the next video. Now I am going to ask one thing. That whatever the here, to better understanding, please go and see the morphology. The flower in the stamens has given detailedly what they given. The sporangia, the whatever the anther is not producing the pollen grain, means sterile, then it is called as, go and see in that one, it is there. And the stamens, what are the filaments are there, that is the filament of the anther, used to fuse with the outer, that is the epipetalus or epipillus. Why it is, that also you have to see. And one more thing, I am asking you one thing. Here it is the bilobe, each lobe toothache. But there is one exam, give the example. I am asking for unilobe and monothecus. Unilobed monothecus. Is there? It is there in your book. He added in the syllabus 2 years back. There the examples are there. That is the Malvaceae family. Remember it. He will ask in that way also. Different lengths of the stamens, cruciferae, and different type of the delphus, monodelphus, didelphus, and polydelphus conditions. All the examples are present. Whatever he has given in the deck. Now we'll see. Whenever we are seeing once more. Here, all the diagram. Sorry, it is my first video I am doing on this digital board. So little bit, but I am doing this one better than the last video, and next video I will become more better. Now you see the thing and read the textbook and the importance of the questions also about sexual reproduction of the flowering plants. Also, I told 6 bits he has given. Previous he used to give 2 to 3 only, but last year he has given 6. Because chances are more to ask twisted questions, statements, matching, assertion, and reason. So many types can be created in the sexual reproduction of flowering plants. So when that much chances are there, they used to prefer such type of the topics. So if you like this one, please like this video, and I am going to do one more video. I will tomorrow I will give one more video, that is the pollen grain development of the, I told microspore will be developed as the pollen grain. How it is, that I am going to discuss. That is very, very important for the NEET. So to see all the things, please subscribe the channel and see this. Okay, thank you.